Hi all. Today we will be discussing the foundations of biological architecture, focusing on carbohydrates, lipids, and how molecular assembly work inside living organisms. Okay, and we will explore how small biological units combine to form large complex structures and how chemical reactions control both the building and the breakdown of these molecules. Okay, now let's see about the type of molecules. Okay. To understand biological molecules, we first need to understand the three important terms that are monomers, polymers, and the macromolecules. Let's start from monomers. So basically, what are monomers? They are simple, irreducible, and biological molecule used as the fundamental building block. Right. So basically, they are small, simple molecule that act as building block. And next we have the polymers. So large molecules constructed entirely of repeating monomer. So as you can see, the structure is repeating, right? And creating a large shape. They are called the polymers. For example, polysaccharides, proteins, etc. So when many monomers join together, they form polymers, which are larger repeating structures, right? Then we have macromolecules. They are overarching category of large complex biological assemblies, right? Which means macromolecules are the broad category of large biological molecules, including carbohydrates, proteins, and lipids. So basically, living organisms create complexity by combining many small units into larger systems. Okay, now let's check the type of reactions. There are two important reactions control molecular assembly, condensation and hydrolysis. Let's start with the condensation. In condensation reaction, molecules join together and release water. This forms bonds such as glycosidic bonds in carbohydrates. Okay, so as you can see here, when the reaction is happening, when two molecules are binding, they will release water, right? And they will form glycosidic bonds by removing the water. Got it? And what about the other one, hydrolysis? Hydrolysis is the opposite process. Water is added to break the bonds apart. Okay, so water will be added to break this bond as separate things. Okay, so these reactions are extremely important in digestion and biological testing. For example, non-reducing sugars must first be hydrolyzed before performing Benedict test, right? We have learned that before. So we have to hydrolyze this. And again, we have to combine this for some other reactions. So these are the diagnostic application. Hydrolysis is a required mechanism to break down non-reducing sugar into reducing sugars before conducting the Benedict test. So that is what you have to keep in mind. Okay. Now let's check the carbohydrates. So basically, carbohydrates are structurally straightforward biological machines, right? Built strictly from foundational sugar monomers, scaling predictably from single unit to massive repeating chains. From single unit to massive repeating chains. So you can see here monosaccharide, disaccharide, and a polysaccharide infinite chain, right? So carbohydrates are considered as true polymers because they are made from repeating sugar units. The structure begins with a monosaccharide, which is a single sugar molecule, right? Monosaccharide is a single sugar molecule. And two monosaccharide combine to form a disaccharide. So two monosaccharides combine to form a disaccharide. And many repeating units join together to form polysaccharides, which are long carbohydrate chains. And finally, we have the polysaccharides. Okay. So this predictable repeating structure is what makes carbohydrates true polymers. Clear? And the base unit, the monosaccharides. So let's see the form. So what is the formula of monosaccharide? C6H12O6. Okay. So monosaccharides are the simplest form of carbohydrate and cannot be broken down further by hydrolysis. So this is the basic unit. Single sugar molecule that cannot be broken down any further by hydrolysis. Okay. For example, we have glucose and fructose. So glucose exists in two forms. First one is alpha glucose and second one is beta glucose. Okay. So alpha and beta glucose are isomers, identical in chemical formula but entirely distinct in physical architecture due to the flipped OH group. Okay. So this is alpha glucose and this is beta glucose. This is how the structure look like. Okay. So it's almost similar, but they have a flipped OH. Okay. So isomer difference, flipped hydroxyl group. Here. So both have the same chemical formula, but the position of the hydroxyl group differ. This small structural difference changes how the molecule behave and what larger structure they can form. So this determines the difference in this structure and its performance. Got it? And now we have to check for the pairs, that is disaccharides, right? So disaccharides are formed when two monosaccharides join together through a condensation reaction, right? When two monosaccharides combine to a glycosidic reaction. So a glycosidic bond is formed. This creates a glycosidic bond and release one molecule of water. So H2O is released. Okay. So these molecules act as intermediate carbohydrate structure before forming large polysaccharides. Okay. What was the examples we mentioned? Sucrose and maltose. And it formed when two monosaccharides are chemically joined via condensation reaction. Okay. So the basic things. And now to the polysaccharides. So polysaccharides, saccharides are the long chain of monosaccharides linked by glycosidic bonds, right? So long complex chains of monosaccharide permanently joined by glycosidic bonds. These represent the highest tier of carbohydrate polymer architecture. So this is how starch will look like. Okay. And this is a structure of glycogen. And this is how cellulose is structured. So they are complex carbohydrate used for energy storage and the structural support. We have examples like glycogen, which stores energy in animals, and cellulose, which forms plant cell walls. So cellulose is the one who forms the plant cell, right? The cellulose. And we have glycogen, which stores energy in animals, right? So it is the energy storage. And this is cell wall. Okay. And these molecules represent the highest level of carbohydrate organization. So these represent the highest level of carbohydrate organization. Now let's see the exceptions. Okay. Lipids. So while lipids are large, highly complex macromolecule, they are fundamentally not polymers. So it's an exceptional case. So they do not contain repeating monomer units. Okay. Instead, their architecture relies on distinct assembly of glycerol and fatty acids. So lipids are different from carbohydrate because they are not true polymers. Okay. So although lipids are large macromolecule, they are not made from the repeating monomer unit. Okay. And instead, lipids are formed from glycerol and fatty acids. This makes them unique compared to carbohydrates and proteins, which rely on repeating structure. So they are not in a repeated structure. Now let's see the comparison. Okay, carbohydrates and lipids. So what is the polymer status? It is a true polymer. It is a non-polymer. And base components are saccharides here, that is sugars. Here it is glycerol and fatty acids. And what is the assembly bond? Glycosidic bond. Here we have ester or something else. And then complexity engine, infinite repeating chains. It is distinct structural variations, right? So carbohydrates are true polymers built from repeating sugar monomers and connected by glycosidic bonds. But lipids, on the other hand, are formed from glycerol and fatty acids and use different type of bonds, such as ester bonds. Even though both use condensation and hydrolysis reactions, their structural organization is fundamentally different. Okay. So they use condensation and hydrolysis, but their architectural blueprints are fundamentally different. So to conclude, biological molecules are built by assembling small unit into larger complex structure, right? So what we have learned so far, first thing, life's physical structure relies on scaling up simple, irreducible building blocks, right? Like monomers into massive biological machines. And carbohydrates are true structural polymers built from repeating sugar units. And lipids are distinct macromolecules that bypasses the repeating polymer rule entirely. And then condensation, that is the removal of water, and hydrolysis, addition of water, act as the universal chemical engine for assembling and dismantling all these molecular architectures. So carbohydrates are true polymers made from repeating sugar units, while lipids are large molecules that do not follow the repeating polymer rule. Finally, condensation and hydrolysis reactions serve as the universal chemical process responsible for building and breaking down this molecular structure. So this is how you have to learn this topic. Thank you.